Restriction enzymes, also called restriction endonucleases, recognize specific base sequences in DNA and cleave both strands at specific places. A restriction enzyme called SAC2 recognizes CCG-CGG, read in the 5' to 3' direction. Notice that a twofold rotational symmetry exists in this sequence. That is, the top sequence and bottom sequence match if both are read in the 5' to 3' direction. In other words, the recognition site is palindromic because it reads the same from left to right as it does from right to left. SAC2 cuts a phosphodiester bond on each strand at a specific cleavage site. The resulting DNA segments in this case have jagged ends with 3' overhangs. Other enzymes may produce blunt ends or 5' overhangs. Restriction enzymes have been purified from a wide variety of prokaryotes, where their biological role is to cleave foreign DNA molecules, providing them with a primitive immune system. Restriction enzymes are indispensable for a variety of laboratory techniques. In one example, they can be used in conjunction with gel electrophoresis to help characterize a sample of DNA. These two DNA segments represent different strains of a virus. One strain has a small DNA insertion. In this procedure, the samples would be incubated with one or more of the enzymes in combination, and then loaded at the top of an agarose gel. DNA is negatively charged, so in an electric field, it migrates toward the anode. Smaller fragments migrate faster than larger fragments. At the end of a run, the DNA pattern in a gel can be visualized by staining the gel with a dye or by using a radioactive probe and the technique of southern blotting. In viral strain 2, some of the fragments are larger than those in viral strain 1. If these sample viruses were of unknown strain type, the pattern of the restriction digest would help identify them.